Hello everyone, my name is AJ Keefe, and in this presentation I'll be covering the paper Autophagy-Dependent Shuttling of TBC1D5 controls membrane translocation of GLUT1 and glucose intake, published in the journal Molecular Cell. So I want to extend a special thanks to the authors for their contribution to this field of research. And for this video, I recommend that my viewers pause the video at each new slide and read through the material and analyze the graphics before playing my commentary. And if you have any questions, please comment them below and I'll be sure to respond. And I hope you enjoy the video. In order to tackle this article, we need to review a couple key concepts. So GLUT1, right here, GLUT1 is a glucose transporter that moves glucose across the membrane into the cell so it can be shuttled into glycolysis. So although GLUT1 is a cell surface receptor, it actually spends most of its time at the endosome inside of cells, down here at the endosome. And during metabolic stress or hypoxia, lack of oxygen, the cell moves GLUT1 from the endosome back to the plasma membrane up here. And this causes increased glucose intake to support an anaerobic glycolysis. Remember, it doesn't require oxygen, and it allows a cell to survive hypoxic conditions. So when a cell doesn't need glucose, it moves the GLUT1 receptors into the internal endosomes. And during glucose starvation, which is right here, we see the recycling transport of these GLUT1 glucose transporters back to the cell surface. So the movement of GLUT1 from the endosome to the plasma membrane is coordinated by this RetroMer complex. So this is the RetroMer complex. It's indicated by this little bar. This is a zoom in of the little bar. You also see the RetroMer complex over here, facilitating recycling. So the RetroMer consists of VPS 35, 29, and 26. It's this endosomal complex that sorts cargo, and it controls the trafficking out of the endosome. So it originally got its name because the RetroMer was observed moving cargo backwards from the endosome to the trans-Golgi network. So this is retrograde transport. So it was called the RetroMer. But more recent research has suggested it also functions to direct cargo from the endosome to the plasma membrane. And some of the things that the RetroMer has been observed transporting includes GLUT1. So GLUT1 is already known to be moved to the endosome and then moved back to the cell surface by this RetroMer complex. So what this RetroMer actually does, though, is it binds to endosomes and then it captures cargo proteins for transport. And GLUT1 would be one of these cargo proteins that's captured by the RetroMer. So think of the RetroMer as a party organizer. It gets everybody together so it can head off to the plasma membrane or the trans-Golgi network. So one of the proteins the RetroMer interacts with and prepares for transport is GLUT1 for the glucose transporter. So the RetroMer coordinates GLUT1 exocytosis from the endosome and moves it towards a membrane. But one big question is, how does the RetroMer get to the endosome and get activated itself? So RetroMer recruitment to the endosome is done by this Rab7 GTPase. So Rab7 is a GTPase, a switch-like protein that alternates between GTP and GDP Rab7. When Rab7 is charged with GTP, it can bind the RetroMer, and so the RetroMer is actually one of its effectors. GTP-Rab7 binds the retromer, it recruits it to the endosome, and the retromer then begins the process of cargo capture and exocytosis. However, and this is really key, TBC1D5 right here, TBC1D5 is a Rab7 gap. It's a guanine activating protein that hydrolyzes Rab7's GTP into GDP. So in other words, TBC1D5 inactivates Rab7 right here, and TBC1D5 inactivates Rab7 by hydrolyzing its GTP, and this prevents the recruitment of the retrosome to the endosome. And without the retromer at the endosome, GLUT1 does not move to the membrane. So the question is, what then coordinates TBC1D5? And we're going to find out in this paper what actually controls 1D or TBC1D5, because this is kind of the deciding factor of whether endosomes are moving to the cell surface or not, because it inhibits the recruitment of the retromer to the endosome. But just to summarize, GLUT1 dynamically shuttles between the membrane and the endosomes, and then back to the cell surface. Rab7 GTP recruits the retromer to the endosome to capture cargo and initiate exocytosis. And this occurs mainly during hypoxia or starvation in order to move GLUT1 back out to the membrane when it's needed. And this Rab7 gap, guanine activating protein, TBC1D5, stops this entire process by hydrolyzing Rab7's GTP into GDP and prevents the retromer's recruitment to the endosome, and thus prevents GLUT1 translocation back to the cell surface. Okay, so we're going to be discussing autophagy, which I'll simply define as a catabolic process of self-eating, literally what autophagy means. Autophagy means self-eating. And it occurs alongside increased glycolytic metabolism and starvation. And autophagy, you might recall, is simply the formation of an autophagisome, you can see right here, around cellular contents that delivers them to the lysosome to be degraded, which then the lysosome will then release amino acids and other basic components used for anabolic processes. And note that in this autophagisome, the autophagisome is marked by the presence of these LC3, which are the green particles or green proteins. So whenever you see LC3, just remember that LC3 is a marker of autophagisomes. And it actually serves as a receptor that binds to cargo to be included into the autophagisome. And since autophagy is a catabolic process that helps a cell cope with starvation and hypoxia, it's not surprising that it occurs alongside increased glycolytic metabolism. And events such as GLUT1 translocation from the endosome to the membrane, this is occurring alongside autophagy. They both are induced by the same glucose deprivation events. So both autophagy and GLUT1 translocation to the membrane are occurring in parallel. But the question is, are these events interconnected? And the answer seems to be yes. So somehow, basically, because previous research has shown that autophagy deficient cells show altered glycolytic metabolism. And this previous research suggested that autophagy directly modulates glycolytic enzymes somehow. But the researchers of this current paper don't buy that explanation. They think there's a deeper relationship and not simply modulating enzymes. So the current paper is designed to investigate other potential mechanisms of how autophagy coordinates or modulates glucose metabolism during hypoxia or during metabolic stress. And so this is what they discovered. These are the answers. So first, autophagy deficient cells during starvation. I forgot to mention that during starvation, autophagy deficient cells during starvation and hypoxia show decreased glucose uptake. They also show decreased lactate production, and lactate is a byproduct of anaerobic glycolysis. And most critically is that there's decreased GLUT1 membrane expression. So when autophagy is deficient, there's decreased glucose uptake, decreased lactate production, indicating decreased glycolysis, and decreased GLUT1 membrane expression. GLUT1 was then observed colocalizing with LAMP1 in autophagy deficient cells, indicating GLUT1 is being shifted towards degradation instead of recycling to the membrane. So during hypoxia or starvation, these autophagy defective cells are actually degrading GLUT1, which is normally needed at the membrane to help facilitate glucose import. So they're literally shooting themselves in the foot. I'm sorry. Then they noticed that in normal autophagy competent cells, the retromer is normally needed to recycle GLUT1 to the plasma membrane. And when the retromer was knocked down in these normal autophagy competent cells, they observed a phenotype that was extremely similar to what they observed in the autophagy deficient cells. And this indicates that the retromer is critical for GLUT1 recycling back to the membrane, which is a finding that's actually substantiated by previous research as well. So the retromer inhibitor, TBC1D5, is the retromer inhibitor, was found to be degraded by autophagy in normal cells to promote retromer-mediated recycling of endosomal GLUT1. So the inhibitor of the retromer is normally degraded by autophagy, and this unleashes the retromer to allow recycling of GLUT1. So when autophagy was defective, the TBC1D5 retromer inhibitor was escaping degradation, and it was constitutively inhibiting the retromer and preventing GLUT1 movement to the membrane. So autophagy is critical to the degradation of the retromer inhibitor and the recycling of GLUT1 back to the surface of these glucose-starved cells. To investigate the effects of autophagy deficiency, they knocked out either ATG5, right here, ATG5 or ATG7 in mouse embryonic fibroblasts. So they started using, the cells they were using were mouse embryonic fibroblasts. And ATG5 and ATG7 are critical components to the formation of the autophagisome. So when they're missing in these knockouts, it basically prevents autophagy. So using these autophagy deficient cells, they decided to begin looking at two basic parameters. They were looking at lactate production in these two graphs and glucose uptake in these two graphs. So lactate production serves as a proxy of hypoxic energy production by glycolysis. And so it's basically a measure of glucose consumption during hypoxia. And 2-NBDG is a fluorescent glucose analog that can be observed entering cells. So these two experiments were aimed at assessing the beginning stages of glucose entering the cell, over here, sorry. So looking at the beginning of glucose metabolism, where it just enters. And then they're looking at the result of glucose metabolism, which is lactate production. So they have the beginning and the end. And what they found was that lactate production was fairly normal in normoxic conditions, so non-hypoxic conditions in the ATG deleted cells right here. We see during high oxygen conditions, there's basically the same amount of lactate production. This is basal levels. What was interesting, though, is that when these ATG5 or ATG7 cells were starved of oxygen, and so they're in hypoxic conditions, the H right here, hypoxic, there's significantly reduced lactate production. So we see about half the amount of lactate that's being produced compared to control cells. What this indicates is that glucose-dependent energy production by anaerobic glycolysis is highly dependent on autophagy, because when autophagy is deficient, we see decreased decreased production of lactate, meaning decreased glucose metabolism. Next, the researchers wanted to see if glucose was even getting into the cells, because obviously a defect in glucose metabolism could simply be a defect in glucose import. I mean, that's kind of the simplest explanation, right? And they discovered that 2-NBDG, the fluorescent glucose analog, was failing to get into autophagy deficient cells. So not so much in ATG5 condition. It's still significant. There is still significantly less glucose getting into the cells during hypoxia. It's just not as pronounced as the ATG7. So in these ATG7 deleted cells, glucose import was severely compromised in both normoxic and hypoxic conditions. So glucose is not getting into the cells when autophagy is deficient, which is by itself a very fascinating observation. Now, I'm personally very curious as to why the ATG5 and ATG7 deletions produce such different results, because it's my understanding that both proteins are equally important to autophagy. So I don't really understand why we would see a difference between these two experiments. But nevertheless, ATG7 deletion did significantly reduce glucose import in both normoxic and hypoxic conditions. So after witnessing and observing such a drastic deficit in lactate production and glucose import, it's reasonable to next examine the expression or the location of the primary glucose transporter GLUT1. So to assay membrane expression of GLUT1 and not overall expression, which wouldn't be very helpful, they use these flag conjugated GLUT1 proteins that are recognized by these very highly specific fluorescent antibodies towards flag. And so what they did is that they pulsed these cells with a fluorescent antibody towards flag on the GLUT1 proteins, and then they washed all of the excess off. So they chased the cells with media not containing the antibodies, and then to analyze the amount of fluorescent antibodies that stuck onto glucose, and they fed the cells into a flow cytometer, and they quantified the amount of surface fluorescence by fluorescence activated cell sorting. So the amount of fluorescence on each cell was measured, and that served as a proxy of GLUT1 surface expression at the time of the antibody pulse. So in panel H, we see pretty clearly that GLUT1 surface expression is normal in both is normal during. Sorry, I'll keep my cursor up here. Flag1 GLUT1 surface expression is normal in normoxic conditions between ATG5 positive and negative cells. So even when autophagy is deficient, we still see normal GLUT1 expression at the surface during normoxic conditions. What's interesting in this panel H, though, is that during hypoxic conditions, during a lack of oxygen, these ATG5 autophagy deficient cells fail to upregulate their GLUT1 surface expression to a significant level. I mean, maybe there's a trend towards increased GLUT1 surface expression, but it's not even significant. In the control cells, the ATG5++, we see a huge, almost 3 times upregulation of GLUT1 at the surface. So the autophagy deficient cells are failing to upregulate their GLUT1 surface expression. And then we can see in the immunohistochemistry itself that in normoxic conditions, so this is normal conditions in ATG plus we see kind of diffuse staining of GLUT1. And in ATG5, we also see kind of diffuse intracellular staining of GLUT1. What's interesting, again, is that during hypoxia and these ATG5 cells, you see lots of staining of GLUT1 along the edges of the cell. So we see lots of surface staining of GLUT1. Now look at what GLUT1's doing in these autophagy deficient cells. It's the GLUT1 expression is inside of the cell. And this is strange because this is hypoxia occurring right here. These cells need glucose, but they're not moving their glucose to the surface. So this is not going to be good for a cell. And we see the same thing right here. The GLUT1 is stuck inside of the cell in these autophagy deficient cells. And then when ATG7 is present, we see lots of GLUT1 at the surface. So autophagy deficient cells are not responding to hypoxia, which normally demands increased glucose import and anaerobic glucose metabolism. But the question is, how do autophagy deficient cells respond to simple glucose deprivation? Well, not surprisingly, they also show severe deficits in the recycling of GLUT1 to the membrane after 2 hours of glucose deprivation. So the white lines in these graphs indicate the autophagy competent cells, and the black lines indicate autophagy deficient cells, either ATG7 deletion or ATG5 deletion. And so a defect in recycling could mean either GLUT1 is not being internalized, or or a general defect in the endocytic machinery. So that they tested these possibilities. So there's a clear decrease in the amount of surface expression of GLUT1 in both conditions. So the question is there a general defect in the recycling of GLUT1, or is it not being internalized in the first place? And so first they looked at the rate of GLUT1 internalization during high glucose conditions. So I don't have this graph shown here, but what they showed is that GLUT1 is internalizing just fine. So during starvation, the autophagy deficient cells can still internalize their GLUT1, during high glucose conditions. So remember, GLUT1 is internalized when cells are fed with glucose. So when there's plenty of glucose around, you don't need GLUT1 importing more. So autophagy deficient cells appear to endocytose their GLUT1 transporters without any problems. So there's no problem there. The next question is general recycling and the endocytic machinery that moves these kinds of transporters around in the cell. And so they actually decided to look at transferrin. Transferrin is a iron transporter that also functions through the pretty much the exact same pathway, the exact same recycling and endocytic pathway, which, like GLUT1 receptors, also moves from the membrane to the endosome and then back to the membrane. And it works just fine in ATG5 deleted cells. And so this is actually a really big point that they're making with this single graph, because what they're saying is that the general pathway, the general endocytic and endosomal transport recycling pathway, is functioning just fine. So even in ATG5 deleted cells, endocytosis recycling is fine. So they're suggesting that it's something specific to GLUT1. So now they're saying autophagy impairs something that's very specific to GLUT1 and not to the general endocytic pathway. So to answer what is specifically wrong with GLUT1, they next decided to look directly into cells and to watch what's happening to GLUT1 when it should have been recycled to the plasma membrane. In order to understand what is going wrong with GLUT1 trafficking, the researchers decided to fuse GLUT1 to GFP so they could literally see it in vivo and to see what compartment the GLUT1 ended up in. So they observed GLUT1 in autophagy-deficient cells, and it was significantly more co-localized with LAMP1. So they found that when they were using a LAMP1 antibody, they saw significantly upregulated co-localization between GLUT1 and LAMP1. So LAMP1 is a marker of the late endocytic and lysosomal compartments. And so this indicates that GLUT1 co-localized with these markers. That indicates that GLUT1 is moving towards degradation, pretty much the opposite path than what it should be doing, because normally GLUT1 is being recycled to the membrane, but in autophagy deficient cells, it's being degraded. So not only is GLUT1 being degraded in autophagy deficient cells, but this colocalization appears to be largely constitutive, meaning in control cells, the amount of colocalization with LAMP1 is kind of dynamic. You can see it decreases during hypoxia or during glucose deprivation, which this makes sense, right? Because you definitely do not want to degrade GLUT1 when you need to import glucose. And you can see control cells, they get that, they understand that. They downregulate the degradation of GLUT1 so they can move it to the cell surface. Autophagy deficient cells, they didn't get that memo because they continue to degrade GLUT1 even when they need to import glucose because they're being starved of oxygen or starved of glucose. So this suggests that autophagy is needed to prevent GLUT1 shuttling to the lysosome and to promote its recycling to the membrane. Now, we know what's happening to GLUT1, it's being degraded, but we don't understand why. So that's what the researchers look at next. So I just had to include this image because you can visually see and appreciate the wonderful immunocytochemistry that they did. So this is kind of just reiterating the last point that we just covered, is that in normal healthy cells, so in these panels, these normal healthy cells, you see the green GFP tagged GLUT1 under basal conditions. It's residing in these endosomes. So in these little pockets, there's a little bit of co-localization with LAMP1, and this indicates that a little bit of the GLUT1 is being degraded during basal conditions. But then you can see during hypoxia and glucose starvation. So during hypoxia or glucose starvation, you see green is lining the surface of the cell because GLUT1 is being transported, and you don't see any yellow. So in other words, you don't see any co-localization between LAMP1 and GLUT because it's not being degraded, and it wouldn't make sense to degrade GLUT1 during hypoxia or glucose starvation. In contrast, you can see autophagy deficient cells, you see tons of yellow. I mean, just look at this glucose deprivation panel right here. Glucose is gone, so they need to import glucose, and look at all the yellow. This indicates LAMP1, GLUT1 colocalization and massive degradation of GLUT1 right when they need it. Not a smart life decision. So at this point, we understand why glucose metabolism is defective in autophagy deficient cells, and that's because they are not importing glucose when they need it, and that's because GLUT1 is being degraded. But why is GLUT1 being degraded? Why is it failing to move to the membrane and instead being degraded? What's responsible for that defect? And the researchers decided to examine the RetroMer complex because it's already known to direct GLUT1 from the endosome to the membrane, and this was confirmed by previous research. And indeed, they found that when they knocked down the RetroMer using shRNA, they knocked down a critical component of the RetroMer, the VPS35. It caused autophagy-competent cells to behave as if they were missing ATG5 or 7. That is, in these autophagy-competent cells, these normal cells, when they knocked down VPS35, it caused GLUT1 surface expression to drop. Sorry, we're looking at these two panels. it caused the GLUT1 surface expression to drop to levels as low as they were in ATG5 and 7 deleted cells. So we can see, here we go, ATG5 ATG5 plus plus VPS35 siRNA. So in these black squares, you can't even see the black squares because they're basically the same as ATG5 deleted cells. So in other words, when the retromer is knocked down, we see the same exact phenotype as we saw in the ATG5 deleted cells. So we also see it in ATG7 deleted cells down here. So in this in panel D, we see not only is GLUT1 surface expression depleted in VPS35 knockdown in these normal autophagy competent cells, but GLUT1 is now co-localizing with LAMP1, just like it did in autophagy deficient cells. We see the correlation coefficient between LAMP1 and GLUT is way upregulated. So this increased correlation coefficient means that GLUT1 and LAMP1 is the overlap colocalization is upregulated when VPS35 is knocked down. So in other words, the researchers found that they could induce this defective GLUT1 sorting in normal healthy cells by knocking down VPS35. This suggests that in order for autophagy to promote GLUT1 relocalization from the endosome to the surface membrane, the retromer complex is required. So the retromer complex is required for autophagy-dependent recycling of GLUT1 to the membrane. How does autophagy modulate the activity of the retromer complex to promote GLUT1 recycling to the membrane? So we learned in the last slide that autophagy-mediated GLUT1 recycling is dependent on the retromer, because if you knocked out the retromer, then the autophagy-competent cells lost the ability to move GLUT1 to the surface and instead began degrading it, just like they did in the autophagy-deficient cells. But we don't know why. We don't know why autophagy is needed to modulate the retromer. So the authors decided to focus first on TBC1D5 simply because it's a known inhibitor of the retromer complex by preventing its interaction with RAB7. And this prevents the retromer from binding the endosome and capturing cargo. So the coup de grace of this paper is in my opinion, it's this single graph here. So shRNA mediated knockdown of TBC1D5, indicated by these black bars, restored GLUT1 membrane expression in autophagy deficient cells. So this graph right here is in autophagy deficient cells. It doesn't show it, but these are. And you can see that when they knocked down TBC1D5, it restored GLUT1 expression. So, I mean, this is pretty huge. That means that TBC1D5 might be responsible for the defects in GLUT1 transport in autophagy deficient cells. It appears that, according to this Western blot to the right, that in autophagy deficient cells, the interaction between TBC1D5 and the retromer is highly upregulated. Okay, so they're doing a co-immunoprecipitation. So we see in these first two columns, these are ATG7 cells. We see that when during controlled basal conditions, immunoprecipitation between TBC1D5 and VPS35, the retromer, is upregulated. This is normal. So we see under normal conditions, the retromer is inhibited by TBC1D5 because they're interacting. When glucose is removed, there's no pull down, there's no association between TBC1D5 and VPS35. So in other words, when glucose is during autophagic conditions, during glucose removal, the retromer inhibitor, TBC1D5, dissociates from VPS35. The really important finding here is that in these ATG7 autophagy deficient cells, the interaction between TBC1D5 and the retromer is actually upregulated during autophagy. And that makes no sense, because this means the retromer is binding and interacting and inhibiting, or getting inhibited by TBC1D5, because we can see increased interaction by this co-immunoprecipitation. So there's actually increased interaction between VPS35 and its TBC1D5 inhibitor during starvation. So the retromer, which is needed for GLUT1 translocation to the membrane, is being stifled by TBC1D5 in autophagy deficient cells. And this caused the researchers to wonder if autophagy was needed to degrade TBC1D5, because that's what autophagy does, is it degrades things. So maybe it degrades TBC1D5, and that would allow the retromer to direct GLUT1 movement to the membrane. So to test this, they examined how autophagy in healthy cells deals with this TBC1D5. So how does it get rid of TBC1D5 in normal cells? So in these Western blots down here at the bottom, again, the TBC1D5 interaction with the VPS35 retromer is downregulated during starvation. So this first graph is just showing us that this is basically the same as before, is that the interaction between TBC1D5 and the retromer is downregulated during glucose starvation. Not too surprising. They then tested the interaction of LC3, the receptor of the autophagosome, with TBC1D5. So they pulled down MYC, MYC, and MYC is fused to LC3A. And it pulled down TBC1D5, indicated by immunostaining towards FLAG, which is fused to TBC1D5. So this is kind of confusing, because they're using two fusion tags. They're using a fusion tag to pull down LC3, and they're using a fusion tag to stain for TBC1D5. So, but just remember that MYC, MYC, is fused to LC3, and FLAG is fused to TBC1D5. But what this darker band right here means, under the glucose deprivation condition, is that there's increased interaction and pull down between LC3A and TBC1D5. So this band right here is critically important for the message of this paper. What it's telling us is that TBC1D5 is being bound and degraded by autophagosomes during glucose deprivation. So that is why the retromer is upregulated in control autophagy competent cells, is because they're getting rid of the retromer inhibitor. So in other words, autophagy upregulates GLUT1 expression at the membrane by selectively degrading the retromer inhibitor, TBC1D5. So let's summarize what we learned in this paper. We learned that defective GLUT1 recycling observed in autophagy deficient cells can be attributed by its mislocalization to the lysosomes instead of its recycling to the membrane. This mislocalization defect is likely a result of retromer dysfunction, because when the retromer is knocked down in normal, healthy, autophagy competent cells, it also causes GLUT1 relocalization to the lysosome. And in these autophagy deficient cells, when TBC1D5 is downregulated by shRNA, it basically restores GLUT1 localization to the membrane. And also, previous research has already shown that the retromer is important for GLUT1 recycling. We also know that autophagy defects don't cause a broad dysfunction in the recycling pathway, because the transferrin recycles without any problems. Instead, autophagy is required for proper TBC1D5 interaction with the retromer, and thus GLUT1 recycling, because autophagy selectively degrades the retromer inhibitor, TBC1D5, during starvation. Thus, autophagy indirectly promotes GLUT1 membrane localization by freeing the retromer from TBC1D5. Given these results, this is what I'm personally left curious about. So how exactly is TBC1D5 being recognized and transferred to autophagosomes during starvation? So the authors observed that TBC1D5 becomes co-localized with autophagosomes, but they don't provide any explanation as to how TBC1D5 might be getting into autophagosomes. And I wonder if perhaps TBC1D5 is being ubiquitinated during starvation and being bound by cargo adapters like P62. And this would trigger recognition by LC3, and it would trigger the incorporation of TBC1D5 into autophagosomes. Another question is, are there any other pathways that might require TBC1D5 degradation and RetroMer activity in order to function? And do they also require autophagy in order to function? So we mentioned that the recycling of GLUT1 is specific to GLUT1, doesn't apply to transferrin. But are there any other pathways that also rely on TBC1D5 degradation and would also be compromised by defective autophagy? Can increased or dysfunctional autophagy from misfolded proteins, such as in neurodegenerative diseases, interfere with the recycling of GLUT1 by downregulating TBC1D5 degradation? So Alzheimer's disease is often called type 3 diabetes, and I wonder if altered autophagic activity could be preventing proper glucose import through this interaction between the RetroMer and TBC1D5. How do inhibitors of autophagy, such as mTORC activation by rapamycin, affect GLUT1 recycling? Does it happen as we might expect based on what we learned in this paper? And lastly, can these findings be replicated at the level of a whole organism? For example, could TBC1D5 knockouts restore glucose consumption in autophagy-deficient organs that perhaps lost autophagic activity by tissue-specific deletion of ATG5? So, for example, we could delete ATG5 in the liver and also delete TBC1D5 and see if maybe that co-deletion would restore glucose consumption in these cells. Anyways, please post any questions that you have, and I'd love to talk about them and we can discuss. And I hope you enjoyed this paper as much as I did. I want to once again thank the authors for this paper, and I genuinely enjoyed reading it, and I think they did a great job. And thanks for watching.